Welcome to the deep dive. Today we're tackling something really crucial in getting new medicines developed. It's called design of experiments, or DoE,
Hmm.
and specifically how it's used in organic process R&D. When you think about making these complex drug molecules, there are just so many variables you could adjust.
Oh, absolutely. Temperature, concentrations.
Right, exactly. It can seem, well, a bit overwhelming. So what if there's a smarter way to handle all that? That's what this deep dive is really about, finding the core ideas behind how DOE helps scientists zero in on the best reaction conditions efficiently.
Yeah, think of it as a peek behind the curtain at how researchers plan experiments strategically. Our mission today is pretty focused, really.
Sure.
We want to explore how DOE is actually applied to optimize those key reaction parameters
Mm.
like temperature, reactant concentration, maybe the amount of a catalyst, the reagent
The
loading.
main levers you can pull.
Exactly. And how varying these, systematically unlocks the best conditions. And importantly, we'll also get into how statistical models are used.
Ah, the stats part.
Yeah, to understand how robust, how stable these processes are, and find those optimal operating windows. We'll touch on examples where lots of conditions are screened too. We're pulling from, scientific papers, standard handbooks, that sort of thing.
Okay, sounds good. So let's maybe start in the beginning. Design of experiments.
Yeah.
What's the basic idea?
Well, at its heart, DOE is just a really intelligent way to plan your experiments. It's different from the old school method.
What, changing one thing at a time?
Exactly. The OFAT, one factor at a time approach.
Yeah,
Tweak the temperature, run the reactions, see what happens. Then go back, change the concentration, run it again.
tedious.
Vary. And you miss things.
Mm.
DOE, instead, sets up experiments where you change multiple factors simultaneously, but in a structured, planned way.
Okay, multiple things at once. How does that help?
Well, the big advantage is you don't just see how one factor affects the outcome. You see how they affect it together, their interactions.
Ah, interactions. Like maybe higher temperature only works well if you also increase something else.
Precisely. That's the kind of thing OFAT often misses completely. DOE is designed to uncover those relationships, so you get a much richer understanding of the reaction.
And presumably you can do that with fewer experiments overall.
That's the key benefit, really. Way more efficient. You get more information, deeper insights from fewer runs, which, for someone trying to get up to speed quickly on this, DOE offers that path to understand these complex relationships without drowning in data from endless single tests.
Makes sense. So how does this look in practice in, say, organic process R&D when chemists are trying to optimize a reaction to make a drug ingredient?
Right. So they have these parameters they can control, yeah. We mentioned temperature, concentration, how much of each region to add, maybe reaction time, even the solvent system.
Lots of knobs to turn.
Definitely. So DOE gives them a framework. It helps them decide which specific combinations of these settings to actually test. They'll choose certain high-low levels for each factor, for instance, and run experiments at specific combinations of those levels.
Not just random combinations, though.
Oh, no, not at all. It's very systematic. By varying these parameters in this structured way across a defined range, they essentially map out the reaction space.
Like creating a contour map?
Kind of, yeah. A map that shows where the peaks are the conditions that give you the best results,
Hmm.
maybe highest yield or highest purity, or minimizing a nasty byproduct, whatever their goal is.
So they use specific experimental plans.
Exactly. There are standard DE designs like factorial designs or response surface designs. Factorials are great for initial screening, looking at the main effects and interactions of several factors.
Okay.
Response surface methods, or RSM, are often used next to really zoom in and find the absolute optimum conditions, like the peak of that mountain on your map.
Huh.
These designs ensure you cover the space efficiently and, crucially, generate data that you can properly analyze statistically.
Right, which brings us back to the statistical models. How do they fit in after the experiments are run?
So you run your designed experiments, you collect your data yield numbers, purity measurements, whatever you're tracking for each run. Then the statistical models come into play. Techniques like regression analysis or ANOVA analysis of variance are used to analyze that data.
What do they tell you?
They quantify things. They tell you precisely how much influence each parameter, temperature, concentration, whatever had on the outcome. Was temperature the biggest driver? Or was it an interaction between temperature and reagent loading?
So they tease apart the different effects.
Exactly. And they mathematically describe those relationships. The models can pinpoint not just the main effects, but also those crucial interactions we talked about. Does changing factor A have a different impact depending on the level of factor B? The model can show you that. It's like decoding the reaction's operating manual.
And this helps understand robustness, you mentioned.
Yes, process robustness. That's a huge concept in pharmaceutical manufacturing. It basically means how well does your process perform consistently, even if there are small, unavoidable fluctuations in conditions.
Like small temperature shifts day to day or slight variations in raw material quality.
Precisely. No real-world process is perfectly constant. So, using DoE and the statistical models, you can figure out which parameters are critical, which ones have the biggest impact if they vary even slightly.
Okay.
And you can define the acceptable operating range for those critical parameters, the sweet spot where you know you'll consistently get good quality product. Understanding this is vital for reliable manufacturing. It gives you that framework for managing variability.
So DOE isn't just about finding the single best point, but also understanding the area around that point where things still work well.
That's a great way to put it, yeah.
Yeah.
Finding the optimum is important, but understanding the boundaries, the operating space where it's robust, is arguably even more critical for large-scale production.
You also mentioned screening studies, where maybe you start with a lot more possibilities. How does DOE help there?
Oh, right. Especially early on, you might have, say, several potential catalysts or solvents or a wider range of temperatures to explore. You need to sift through these efficiently.
Kind of like high throughput screening.
Well, HTS is often about testing thousands of discrete compounds quickly. DOE in process chemistry is more about strategically exploring continuous variables like temperature or concentration or combinations of factors. Instead of trying, every single possible combination, which would be impossible, DOE lets you select a smart, smaller set of experiments.
A representative sample of the possibilities.
Exactly. A well-designed screening experiment using DOE principles can efficiently highlight the most promising factors or regions in that vast parameter space. It guides you.
So it points you in the right direction.
Yeah. You run the initial screening DOE, analyze the results, and it tells you, okay, this catalyst looks best, or focus your efforts in this temperature range. Then you can follow up with more detailed optimization DOE studies within that narrowed down space. It's a much more efficient staged approach.
It really does sound like efficiency is the watchword here.
Oh, absolutely. That's one of the biggest drivers for using DOE.
Yeah.
You maximize the learning from every single experiment. You explore the reaction possibilities systematically and uncover those interactions.
Which ultimately saves time and resources.
Tremendously. Faster process development, less waste of expensive materials, quicker path to finding a robust manufacturing process. In pharma, where time to market is so critical, these efficiencies are invaluable. It means getting important medicines to patients sooner.
Okay, so let's try and pull this together. What we've really seen is that design of experiments is this powerful, systematic method used in organic process R&D. It's about cleverly planning experiments to efficiently find the best reaction conditions, optimizing things like yield and purity by looking at factors like temperature, concentration, and so on.
Right.
And crucially, it uses statistical models to understand not just the main effects, but the interactions between factors, and to figure out how robust the process is, defining those safe operating ranges.
You got it. And it helps screen possibilities efficiently, too.
Yeah, getting the most knowledge from the fewest experiments, essentially. It provides that structure to avoid just random trial and error.
That sums it up very well. It really provides those aha moments, understanding the logic and efficiency behind optimizing these complex chemical processes.
It definitely does. It's quite illuminating to see the kind of structured thinking involved. And it makes you think, doesn't it?
Hmm.
These core ideas, systematically testing variables, understanding interactions, using data to define robust operating zones. I wonder how applicable that kind of thinking is to other complex problems we face, maybe outside of chemistry, in, business processes, or even just everyday troubleshooting.
That's a really interesting thought. The principles are quite general, aren't they? Systematic exploration and data-driven understanding. And hey, if this deep dive sparked your interest in the chemistry side,
Yeah.
I really encourage listeners to look into some actual case studies or scientific papers on DoE and pharma development. There's a lot more fascinating detail out there if you want to go deeper.